All right, we'll go ahead and get started. Good morning and good afternoon, everyone. Welcome to the University of Arizona Food Safety Consortium Summer Seminar Series. I am Sadhna Ravishankar. I am a professor at the School of Animal and Comparative Biomedical Sciences at the University of Arizona and also the chair of the Food Safety Consortium. First of all, I would like to thank our speaker today for giving us the presentation. And I would like to thank you all for joining us today. And before I introduce the speaker, a few announcements. We do have a general monthly meeting of the Food Safety Consortium on the 24th of August between 2 and 3 p.m. So, please join us if you can. That's the next event. And the Food Safety Conference would be happening sometime in October. Once we have a date finalized, we will inform you. And in today's seminar, we will have initially the presentation, and then we will have a Q&A towards the end. And please post your questions in the chat area, and I will read out the question for the presenter so that she can answer the questions. So it is my pleasure to introduce today's seminar speaker, Dr. Hsin-Bai or Amy Yin. She is currently a postdoctoral research associate in Dr. Jeetu Patel's lab at the Environmental and Microbial Food Safety Laboratory, USDA in Beltsville, Maryland. Amy received her bachelor's from the National Chiayi University in Taiwan in 2010 and a PhD in animal science from the University of Connecticut in 2017. Her doctoral research focused on investigating the efficacy of natural phytochemicals in protecting chickens from salmonella colonization and aflatoxin toxicity. Her postdoctoral research focuses on identifying the microbial contamination of fresh produce via irrigation with alternate irrigation water sources, as well as developing natural antimicrobial strategies to improve microbial safety of fresh produce at both the pre- and post-harvest levels. Dr. Yin has published 30 peer-reviewed manuscripts in professional scientific journals and 5 book chapters. And she has given more than 20 presentations to the at professional conferences. Today, she is going to talk about a very, very important topic that is of concern to the food industry, biofilms. She's going to talk to us about the research she has done in Dr. Jeetu Patel's lab on biofilm formation and control strategies in food processing facilities. So welcome, Amy, and it's all yours. Thank you again.
Okay, hello everyone. Thank you for the introduction. I'm very honored and very happy to be here, and then thank you for having me. So in today's presentation, I'm going to share the biofilms related works that conducted in the USDA ARS laboratory, leading by Dr. Jitendra Patel. And also, I have also summarized some of the recent publications that focus on the controlled biofilm strategies. So with that being said, the title of my presentation today is the Biofilm Formation and Control Strategies in Food Processing Facility. So many of you may have already known that pathogenic and spoilage bacteria biofilms in industrial environment could be a serious problem because bacteria in the biofilms become more resistant to the disinfectant. So they can survive after the disinfection procedure and remain in the environment and could potentially cross-contaminate the food products processed in the food processing facility and eventually causing the foodborne human outbreaks. So in the food industrial settings, biofilms may be formed on abiotic and biotic surfaces. Food contact surfaces where the food has been processed on provide an excellent niche for the bacteria to survive and form biofilms because because of their nutritional richness. At the same time, biofilms can also be found on the equipment surfaces where, especially for those surfaces with high humidity and moisture content, such as the washing tank. So to give you a very brief introduction about biofilm formation in the food processing facility may be different from their natural ecosystem because of the nutritional richness in the food processing facility. So the biofilm formation process begins with a reversible attachment where cells loosely attach to the surface, followed by an irreversible attachment where cell to surface adhesion has been enhanced. And then cell multiply and form small aggregates of bacteria called microcolonies. When conditions are sufficient for enough growth of microcolonies, biofilms start to develop an organized three-dimensional structure. The final stage of the biofilm formation is called dispersion. It's when bacteria inside the biofilms produce enzymes that break the biofilms and release the bacteria back to the environment and start colonizing the new surfaces. So biofilms can cause serious food safety problems in different food processing facilities. For example, in the dairy industry, even though there is a pasteurization process, but spore-forming bacteria such as Bacillus cereus can survive this process and could become a great concern in the dairy industry. Bacteria from the poultry carcasses can also be transferred from the meat to the equipment surface and cause cross-contamination. Chicken meat and eggs are frequently associated with severe foodborne outbreaks, especially for Salmonella enterica. In the red meat industry, bacteria from the red meat can form strong biofilms on the equipment surface, especially for the stainless steel. And Shiga toxigenic E. coli, especially for serotype 0157H7, is frequently associated with red meat industry outbreaks. In addition, Listeria monocytogenes is another pathogen will be able to contaminate ready-to-eat meat. One of the most commonly found bacterial species in the seafood industry is Vibrio species. Other bacterial pathogens such as Salmonella enterica and Listeria monocytogenes are also associated with the formation of biofilm in the seafood industry. Fresh food and vegetables have also the potential to harbor bacterial pathogens, including Salmonella, Aztec, and Listeria monocytogenes. And it has been reported that most of the bacteria isolated from the fresh cut facility are able to form biofilm. And colonization of fruits and vegetables can occur at any stage during the pre and post-harvest operations. So several factors that could affect the bacterial attachment and the subsequent biofilm formation. Surface property of bacteria such as the surface protein, including capsule, flagella, surface charge, or hydrophobicity of the bacterial surface will affect their initial attachment onto the surface. Strain variation could be another bacterial factors. For example, study has shown that plant-associated E. coli isolates have the ability to produce more biofilms as compared to the isolate from human and other mammalian hosts. The physical characteristics of the abiotic and biotic surfaces in the food processing facility also are critical for biofilm formation. So some commonly used materials such as stainless steel and plastic will become more rougher and rougher after several times of the reuse. And then studies have already shown that bacteria could be more easier to attach and contaminate the surface once become rougher, so making them more susceptible for biofilm formation. And some of the environmental factors, including nutrient composition, temperature, pH, and osmotic pressures, will all affect the biofilm formation. So let's look into more details for the effect of bacterial characteristics on the biofilm formation. For example, curly fimbriam is a fibrous surface protein that is important for biofilm formation by E. coli and Salmonella. One of our previous publications by Dr. McCarrison in our laboratory used curly deficient E. coli strands to determine whether this curly protein is important for bacterial attachment onto the spinach surface. And as you can see, the curly deficient strand was not able to produce a red phenotype on the Congo red agar as compared to the wild type. So the results of our study observed strong attachment of E. coli to spinach surface during the 40 hours of incubation after inoculation is greatly influenced by their ability to produce curly. So as you can see, the curly deficient strands circled in blue color had a significantly lower attachment and persistence of E. coli populations as compared to the wild type strain. So the next step of our study, we further investigated the effect of spinach cultivar on the bacterial attachment. So on the left-hand side, you can see the scanning electron microscopy images showed 4 different cultivars used in this study, including Amelia, Lazio, Space, and Wataki cultivars. They all had different surface characteristics. And the figure underneath here showed the quantified leaf roughness and the number of the stoma. And then you can see that Amelia cultivar had the least leaf roughness and least numbers of the stoma. On your right-hand side showed the result of 3 different strands. The top of one showed the wild type, and the bottom 2 figures show the mutant strand, which was the curly mutant or the double mutant strands. So that's focused on the top figure here. By using the wild type, you'll see that after 14 days of the inoculation, the recovery of E. coli from Amelia cultivar became undetectable as compared to approximately 3 log CFU per gram of recovery from other 3 types of the cultivars. And as I just mentioned a few seconds before, Amelia happened to be the cultivar had least leaf roughness and least numbers of the stoma. And the cultivar effect was more pronounced when we used curly deficient strands. As you can see for the figures here on day 7, the recovery of the E. coli populations from the Lazio and Wataki cultivars were significantly higher than the other 2 cultivars. And if you take a look at the figures here, the Lazio cultivar had the greatest number of the stoma, and the cultivar Wataki had the greatest of the leaf roughness. In the same study, we visualized the attachment of E. coli onto spinach by using the confocal fluorescence microscopy. Results reveal that the curly expressing strands attached in greater numbers, showing in green colors as compared to the curly deficient strands. And the highest density of bacterial cells were observed at the stoma poles and at junctions between epidermal cell walls at all time periods after inoculation. Another study published recently from our laboratory has also shown the bacterial strand variation and the type of the surface on the biofilm formation. So we have used different strands for each bacterial species. As you can see, there are 3 different strands for L. mono, 5 different strands for E. coli 0157H7, and 3 strands for non-0157S-Tec. And the biofilm formation were determined on 2 different surfaces. The black bar showed the polycarbonate results, and the other one showed the stainless steel results. And as you can see, that's focused on the E. coli 0157H7 now. Among the 5 different strains that have been tested, we have found a relatively stronger biofilm former on both surfaces for around 6 log recovery as compared to a relatively weaker biofilm former at 4.5 log recovery. And also, we have found that the recovery of E. coli populations from the polycarbonate surfaces was approximately 1 log higher than the recovery from the stainless steel surface. So besides the researchers that focused on the biofilms formed by single species bacteria, microorganisms most likely exist as a complex community consisting of several species at the food processing facility. And several conditions in bacterial biofilms could be altered between the single and multi-species biofilms, including the enhanced production of EPS, physiological status of the bacteria, interspecies cross-protection, internalization into the food. And bacterial interactions in multi-species biofilms can also be categorized as competitive, cooperative, or neutral. So on the right-hand side here showed one of the examples as the competitive bacterial interaction in multi-species biofilms. So a publication showed that biofilm formation of Listeria monocytogenes was decreased when co-cultured with a lactic acid bacteria called Lactobacillus paraplantarum due to the production of antimicrobials such as lactic acid and bacteriocins. Bacterial interactions can also be categorized as cooperation when all the microorganisms in the community are benefited, and most of the time will lead to increased biofilm formation. For example, Liu et al. In 2016 investigated the effect of Ralstonia insidiosa on the biofilm formation by 3 different pathogens, including Listeria monocytogenes, Salmonella enterica, and E. coli. And results of their study showed that Ralstonia insidiosa facilitated the biofilm formation by all 3 pathogens that were tested because Ralstonia is highly efficient for nutrient utilization. Another example of the cooperative interactions is the increase of biofilm biomass by 2 different serotypes of the E. coli, published by Zhang et al. In 2020. As you can see that the dual strands of E. coli biofilms has been increased as compared to single-strand E. coli biofilms, and the ratio showed 70% of the E. coli are from E. coli 091 and 30% from 0157. So the organization of microorganisms within multi-species biofilms is not random, but followed a specific pattern that contributes to the fitness of the whole community. Generally, there are 3 different types of the bacterial organization patterns. The first one is called separate single species microcolonies, which refer to an organization where each species form a separate microcolony side by side, which result in a neutral bacterial interaction. Alternatively, biofilms can also have a structure where species are mixed together throughout the biofilms, which is referred as intermixing. Finally, biofilms can also have layered structure, where one species is located in the lower layer and the other species is located in the upper layer. And this type of the organization pattern can result in both either cooperative or competitive bacterial interactions. And I just want to point out here before I switch to the next topic that even though I just say that multi-species biofilms is most commonly found format in the food processing facility, but multi-species biofilms is considered a complicated community to be studied. And even though you may find a few publications that put the effort to study the bacterial interaction for multi-species biofilms, but it is still poorly explored as of now. So in the following slides, I would like to share with you some of the commonly used strategies for biofilm control in the food processing facility, as well as some of the emerging anti-biofilm strategies. Regular cleaning and disinfection procedures are important in the industry to prevent biofilms. Depending on the specific industry process, cleaning and disinfection on the whole infrastructure is possible to constantly avoid the bacterial contamination on the pipelines and surfaces. Alternatively, clean-in-place methodology can also be done to maintain the clean surface by spraying and recirculating the disinfecting liquids. Currently, the physical methods, including hot steam and ultrasonification, and some of the chemical-based methods, including quaternary ammonium compound, chlorine disinfectant, sodium hydroxide, and parasitic acid, are nowadays being used in the food processing facility for biofilm control. Hypochlorite is one of the most commonly used sanitizer in the food industry due to their high efficacy and low cost. Parasitic acid is another sanitizer that is being very popular being used in the food processing facility. However, these sanitizers may cause corrosion on equipment surfaces, and they can also pose risk to the workers. Alternatively, quaternary ammonium compounds can also be used. They are relatively harmless to the equipment and also pose less risk to the workers. However, this compound is not as effective as compared to chlorine and parasitic acid. So because of the limitations of these commonly used chemical-based strategies nowadays, lead to the development of the new strategies for controlling biofilms. And these approaches must be developed that avoid bacteria to build resistance to the disinfectants. They should also be effective, safe to be used, easy to apply, and cost-effective. So here, three categories of the novel natural approaches against bacterial biofilms are listed here, including the non-toxic antimicrobial, the use of protecting cultures, and the involvement of the new technology. So first, let's focus on the use of non-toxic antimicrobials. The use of natural antimicrobial agents has been increasing, not only due to the concerns for the emergence of the antibiotic or antimicrobial resistance, but also due to the consumers' awareness for healthier and non-synthetic ingredients for their food and disinfectant. The major sources of phytochemicals are aromatic or medicinal plants. Other sources of natural antimicrobials include vegetable byproducts such as seed and peels of the fruits and vegetables. Numerous phytochemicals, as you can see from this table here, have been successfully evaluated as anti-biofilm agents in different applications. Besides to their generally recognized as safe status, these phytochemicals have different mode of actions, making them more effective and potentially possibly overcome the problems of antibiotic or antimicrobial resistance. Take one of the most recent publications as an example for evaluating the phytochemical as antibiofilm agent. Green cardamom oil is an essential oil for flavor enhancer and preservative in food products. And the authors have first tested the GCMS analysis to determine the major bioactive compounds in the green cardamom oil. And after that, several different concentrations of the green cardamom oils were used to inhibit the biofilm formation by E. coli 0157H7 and Salmonella typhimurium. And crystal violet method was used to quantify the biomass of the biofilms formed by these pathogens. So the results table here, as you can see, the concentrations of green cardamom oil between 0.015 to 0.125% reduced biofilm formation by approximately 64 to 85% by E. coli. And these concentrations also inhibited 45 to even 100% of the biofilm formation by Salmonella. One of our lab's previous publications reported the removal of biofilms by two phytochemicals, namely cinnamomydehyde and sporin. Sporin is a mixture of eugenol, thymol, and rosemary extract. So as you can see, different serotypes of the Salmonella and also different strands were used to form biofilms on the polystyrene surfaces during 48 hours of incubation. And after that, these contaminated surfaces were treated with 30 minutes of different concentrations of the cinnamomydehyde at 0.1 or 0.2%, or sporin at 0.1 or 0.2%. And as you can see that after 30 minutes of the contact time, there was approximately 2 to 6 log reduction of Salmonella populations in biofilms as compared to the untreated control. Another group in the non-toxic antimicrobials is called peptide. Short-length peptide that contains 15 to 30 amino acids with antimicrobial activities are commonly named as antimicrobial peptides. One of our previous publications studied the efficacy of an antimicrobial peptide called peptide 1018 for removing biofilms on equipment surfaces. So different strands of the Shiga toxigenic E. coli and Listeria monocytogenes were used to form biofilms on polycarbonate and stainless steel coupons in a CDC bioreactor for 40 hours incubation. And after 48 hours, the coupons were treated with different concentrations of the peptide 1018 solutions ranging between 10 to 50 microgram per ml. And after 24 hours of the treatment time, surviving bacterial populations in biofilms were determined. So as you can see the results, the top one showed the result of L. monocytogenes and the bottom one showed E. coli 0157H7. And as you can appreciate, we observed an concentration-dependent antibiofilm activity of peptide. As you can see, the reduction has been increasing while the concentration increased from 10 to 50 microgram per ml. And overall, we found a 4 to 5 log reduction on the stainless steel after 50 microgram per ml of the treatment concentration for Listeria monocytogenes, and a 1 to 3.5 log reduction for E. coli 0157H7 on the stainless steel after treated with 50 microgram per ml of the peptide 1018. So in the next group of non-toxic antimicrobial, acids have been tested as potential antibiofilm agent. Liu et al. In 2017 studied the efficacy of a phenol-lactic acid, which is the acid that could be produced by lactic acid bacteria for removing Listeria monocytogenes biofilms. So Listeria monocytogenes was used to form biofilms on polystyrene surface under different incubation conditions. It was either at 37 degrees Celsius or at 15 degrees Celsius for up to 7 days. And after that, the polystyrene surface were treated with different concentrations of the phenol-lactic acid ranging between 0.25% to 3% for a total of 5 to 16 minutes exposure time. So the results here, as you can see, that the antibiofilm activity of phenol-lactic acid also showed a concentration-dependent manner. The concentration increased from 0.25% to 3%, showed the enhanced antibiofilm activity, and also increased when the exposure time increased from 5 to 16 minutes. Overall, these phenolactic acid concentrations at 1% and 3% showed a effectiveness for controlling biofilms by Listeria monocytogenes just for 5 minutes of the exposure time. So the next group of the emerging natural approaches, I would like to talk about the protecting cultures, including lactic acid bacteria and bacteriophages. Lactic acid bacteria can be isolated from various sources, including from the food products, and may have a probiotic characteristics. In addition, lactic acid bacteria can also produce antimicrobials, including organic acid, hydrogen peroxide, and also bacteriocin. Bacteriocin produced by lactic acid bacteria are known to target closely related microorganisms, including pathogens. Among the scientific methods, there are different ways that the lactic acid bacteria and bacteriocin work against pathogen biofilms, including first one was the displacement, is when the lactic acid bacteria and bacteriocin work together against the pathogens that have already formed biofilms on the surface. The next one is called exclusion, is when the pathogens were introduced to the surface that have already been covered with the lactic acid bacterial biofilms. The third one is called competition, is when pathogen and lactic acid bacteria were co-cultured together to have a competitive bacterial interactions. So then the initial attachment of the pathogen to the surface will be inhibited. The last one is called inhibition. It's for the bacteriocin to work alone to inhibit the pathogen attachment onto the surface. Lactic acid bacteria and bacteriocins are attracting serious attention in the recent years and are extensively studied for their potential applications against biofilms. As shown, this table summarizes the most recent 5 years publications that use lactic acid bacteria alone or in combination with bacteriocins through different scientific methods, as I just mentioned before, for different targeted pathogens on different equipment surfaces. So this study is an example of the competition effect of lactic acid bacteria against pathogens for biofilm formation. 6 isolates of lactic acid bacteria were isolated from kimchi, and they were used to co-culture with Listeria monocytogenes. And on the stainless steel coupons, as you can see, when the Listeria monocytogenes was co-cultured with different strands of the lactic acid bacteria, the biofilm formation has been significantly reduced as compared to the untreated control. And similar results has also been found on the latest leaves, where when the Listeria monocytogenes were co-cultured with lactic acid bacteria showed approximately 1 log reduction as compared to the untreated Listeria. And the confocal microscopy images also supported their population results. As you can see, the biofilms developed by Listeria alone had more live cells indicated in green color as compared to when Listeria was co-cultured with lactic acid bacteria. The biofilms has been damaged with more dead cells indicated in red colors. The next study used only bacteriocins produced by lactic acid bacteria against biofilm formation by pathogen. And in this study, they used a sub-inhibitory concentrations of the bacteriocin called BM173. The sub-inhibitory concentrations are the concentrations that does not affect the growth of the pathogen, to ensure that the biofilm inhibition ability observed was not solely due to the reduced populations of the pathogens. So in this study, sub-inhibitory concentrations of BM173 were treated with E. coli or Staph aureus for 24 hours at 37 degrees Celsius, and crystal violet method was used to determine the biofilm formation. And for the E. coli, as you can see, when E. coli was treated with sub-inhibitory concentrations of the BM173, there was a 40 to 70% of the biofilms was inhibited as compared to control. Likewise, there was a 20 to 55 inhibition for biofilm formation by Staph aureus as compared to the untreated control. Bacteriophages are another alternative for development of sanitizers. Phages are bacterial viruses with high specificity to the target bacteria, and they are harmless to humans and are abundant in the environment. They have the ability to penetrate into the inner layer of the biofilms to inactivate the biofilms to inactivate the bacteria inside the biofilms, unlike other commonly used chemical sanitizers whose penetrating ability is quite limited. So this table summarizes some of the recent publications that use the commercially available bacteriophages or the environmental origin bacteriophages for different target bacteria under different applications. Host range is considered to be an important phage characteristic, and phages with wide host range but with high specificity are usually more preferred. A study done by Zhang et al. In 2020 used a bacteriophage called FP43 for controlling pathogenic and non-pathogenic E. coli biofilms. So the first test they have did is to determine the host range. And as you can see that FP43 can successfully lyse the both non-pathogenic and pathogenic E. coli, including several different serotypes of the Shiga toxigenic E. coli, showing it had the wide host range, and at the same time also showed high specificity because it did not lyse other non-E. coli bacterial pathogens. So further, this FP43 was tested for its ability to inhibit biofilm formation and for removal of biofilms. So on your left-hand side, showing the result of the inhibition of the biofilm formation. So equal numbers of the two different strands of the E. coli were added and incubated with or without FP43 for 48 hours. And as you can see that the biofilm formation was inhibited as compared to the untreated controls and the presence of the FP43 throughout the entire 48 hours incubation. And by the end of 48 hours incubation, there was a 41% reduction for biofilm formation as compared to the control. And at the right-hand side, show the removal ability of FP43. So the polystyrene surfaces that have already formed with the dual strands E. coli biofilms were treated with FP43. And as you can see that within 6 hours of the treatment time, there was a 50% eradication of biofilms being observed as compared to the control. And this number did not increase throughout the entire 48 hours incubation. Another study published by Wang et al. Evaluated the effect of another bacteriophage called AZO145A against Shiga toxigenic E. coli. And as you can see on the left-hand side, also showed the host spectrum of this bacteriophage, showed it has a wide host range for different serotypes of the E. coli and did not lyse Salmonella here. And on the right-hand side, show the removal of this phage. Regardless, the the age of the biofilms was at 24, 48, or 72 hours, the treatment of azole 145A phage for 3 hours resulted in a 2 to 3 log reduction of bacterial populations in biofilms as compared to the control. And the microscopy images also supported that the results showed that less aggregate cells were found in the presence of the phage treatment as compared to the control here. So the next step of this study, Wang et al. Investigated the transfer of the E. coli from the contaminated stainless steel coupons to the beef. So stainless coupons with 40 hours old E. coli 0145 biofilms were treated with or without bacteriophages for 3 hours, and then they are used for two different scenarios. One is called slide method, is when the beef pieces was used to slightly touch on each side of the contaminated stainless steel coupons that were treated with or without the bacteriophage for 10 seconds. And the sandwich method was used for two individual beef pieces that touch on each side of the contaminated coupons, and they used a glass bottle on the top to provide a consistent pressure for 10 minutes. And the results here, as you can see that results prove that the stainless steel coupons that has been contaminated with E. coli biofilms can be very easily transferred to the beef, regardless that was like a 10 seconds contact time or 10 minutes contact time, all resulted in 3 to 4 log CFU per coupon of bacterial populations on the beef. And when the stainless steel coupons were pre-treated with phage, the transfer of bacterial populations reduced to 0.5 log CFU per coupon. Another publication from our laboratory used the environmental E. coli 0157 phages to kill the bacteria that attached onto the blades used in the spinach processing facility. Under different experimental incubation conditions, approximately 5 log CFU per blade of E. coli 0157H7 were found to attach onto the blades. And these contaminated blades were further treated with a cocktail of E. coli specific bacteriophages for 2 hours through spraying treatment. And after 2 hours of the treatment time, we observed a greater than 4.5 log reduction on the blade for the bacterial populations attached onto the blades, which become undetectable, which is less than 0.4 log CFU per blade. So the final portion of my presentation today, I would like to introduce you the involvement of the new technologies with the biofilm control approaches. Nano-emulsions are formed by mixing oil, emulsifier, and water, and they are optically transparent or semi-transparent. Because nano-emulsions are very simple in terms of their composition, the toxicity of the nano-emulsion is expected to be low. Therefore, they are very easily to be incorporated in food-related applications. There are two different methods that can generate the nano-emulsions. The high-energy methods use the mechanical devices that generate the intense interactive forces that break up the water and oil phases. Cinnamic acid, along with other essential compounds such as cinnamaldehyde and curcumin, are one of the most studied phytochemicals in nano-based technology for biofilm disruption. In this study, Ladicity et al. In 2018 used tranexamic acid nanoemulsion as anti-biofilm agent against 3 different pathogens on polystyrene surfaces. And as you can see, the results here show that the left figure showed the result of using the cinnamic acid nanoemulsion. on the right-hand side shows the use of pure cinnamic acid oil. So as you can see that the concentrations that required to inhibit 50% of the biofilm formation for for Staphylococcus was at 0.1 milligram per ml for when used nano-emulsion. However, this concentration was at 0.39 milligram per ml in order to inhibit more than 50% of the biofilm formation by using the oil format, not the nano-emulsion. And the similar trend has been observed for rest of the two pathogens that the concentrations required to inhibit 50% of the biofilm formation was always higher when using the pure oil format as compared to the nano-emulsified format. Similarly, for next one, show the removal of using tranexamic acid nano-emulsion. And again, as you can see, the concentrations that required to eradicate 50% of the biofilms formed by these pathogens are always lower when used nano-emulsion as compared to using tranexamic acid or pure oil format. One of our lab's current under review paper reported the use of carbocore nano-emulsion as a post-harvest washing treatment against E. coli 0157H7 on fish produced. Carbocore is a phytochemical derived from oregano oil. And the plant leaves that were inoculated with E. coli 0157H7 were treated with water alone or chlorine at 25 or 50 ppm, or carbocore nano-emulsion at 0.25 or 0.75%, and stored for 14 days at 10 degrees Celsius. And as you can see on day 14, you can start seeing the development of the bacterial aggregation and indicating the biofilm formation on the plant leaves that were untreated or treated with water alone, or even with the chlorine treatment, you can see the aggregation of the E. coli cells on the plant leaves. However, when the plants were treated with carbocore nano-emulsion, you can only see individual scattered cells on the plant leaves, indicating that the biofilm formation has been inhibited. So phytochemicals have also been encapsulated in metal-based nanoparticles. Metal nanoparticles are produced from the reduction of metal ions such as gold, silver, copper, and zinc, which often employs with a microbial or plant extract to create non-toxic materials. Ramasamy et al. In 2017 used a cinnamaldehyde encapsulated gold nanoparticles as anti-biofilm agent against E. coli and Staphylococcus on polystyrene surfaces. And as you can see that different concentrations of the cinnamaldehyde gold nanoparticles significantly inhibited the biofilm formation for all different pathogens that has been tested and showing in a concentration-dependent manner. And this result can also be obviously observed under the fluorescent microscopy images, as you can see that the biofilm formation are significantly inhibited when treated with cinnamaldehyde gold nanoparticles as compared to using cinnamaldehyde pure oil alone or the untreated controls for all 4 different pathogens. Nanobubbles are tiny gas bubbles that are thermodynamically stable for many months or even longer. The bactericidal properties of nanobubbles are similar to ozone, which is an oxidizing agent. So one of the recent publications suggested the use of treatment of nanobubbles and electrolyzed water alone or in combination to control E. coli 0157H7 biofilms on stainless steel surface and plastic surface. And as you can see that nanobubbles induced 3 to 5 log reduction in E. coli biofilms on stainless steel surface within 2, 5, and 10 minutes of the treatment time, and also reduce a 2 to 3.5 log reduction on plastic surface. When the electrolyzed water was introduced with nanobubbles, that resulted in a completely eradication of biofilms on the plastic surface. That's more than 8 log reduction for 5 and 10 minutes treatment time, as well as on the stainless steel surfaces. So the final part of the involvement of the new technology is the engineered surfaces. In general, coatings that are able to prevent biofilm formation can be classified as either antimicrobial coatings or anti-fouling coatings. The antimicrobial coatings can kill bacteria when they approach to the surface by releasing the antimicrobials. And anti-fouling coatings will avoid the bacterial accumulation on the surface through their repellent properties. So they could be either hydrophilic or superhydrophobic surfaces. So Ben et al. In 2020 fabricated the nano-engineered stainless steel surfaces by using electrochemically etching and surface coating as anti-biofilm surface against E. coli 0157H7 and Salmonella. The stainless steel surface was electrochemically etched for 5 to 15 minutes, followed by addition coating of 0.2% of the Teflon to create a super hydrophobic surface. So as you can see for the attachment, showed that bacterial attachment onto the electrochemical etched surface was significantly inhibited as compared to the untreated surface, and the further coating further reduced the attachment of pathogens as compared to two other surfaces. Similar result has been observed on the biofilm formation. The etched and Teflon-coated surface had the least biofilm formation as compared to other two surfaces. And it also can be very obviously observed under the microscope to show that if there is a dense biofilm formation on the untreated surface as compared to the etched and Teflon-coated surface. So today's presentation summarized that the most recent published works focus on the use of natural anti-biofilms approaches, and some of them have the involvement of the new technologies. These methods are mostly effective and safe to be used for biofilm controls. However, factors that affecting their effectiveness cannot be underestimated, such as I just mentioned about the single versus multi-species biofilms, and all other factors, including environmental factors, bacterial characteristics, and the type of the first surfaces will all affect their effectiveness. The next generation of the nanomaterials could be the one that combine with the existing evidence and then increase their applicability. So with more and more studies that are standardizing the manufacturing protocols, it is promising that a new smart natural approaches will be made in the nearly future that could have a significant impact on biofilm control in the food processing facility and protect the public health. So right now, I would like to thank my supervisor, Dr. Jitu Patel, for all his support and guidance, and my strong team members, Chi-Hong, Ashley, Grishma, and also our previous postdoc, Dr. Shiva Kirlad and Dr. Demetru Makarasen. Also, I would like to thank our collaborators on the Nano Emotion project from Dr. Lord's lab and from University of Connecticut, Dr. Abhinav Upadhyaya. So with that, I would like to conclude my presentation today, and thank you for your attention, and I'll be happy to answer any questions.
Thank you, Amy, for the wonderful presentation. Lots of useful information regarding biofilm prevention and control. So if anybody has any questions, please submit them via chat, or you can even unmute yourself and you can talk. So to begin with, I have a couple of questions. The first one is, I know you talked about using essential oils and active components. What are the concentrations you used? And did you see any changes in the surface of the material, such as polycarbonate plastics, different types of plastics that you used with repeated use at these concentrations? Did you see any changes in the material?
So the concentration we use is quite low. As you can see, it's only like 0.1 or 0.2%. So for that low concentrations, we did not see any effect on the surface. Of course, in terms like a long-term use, we probably have to do more research, like if that's been repeatedly used, if that will affect the surface. But I would expect that won't change anything on the surface because again, the concentration is very low. And the concentration we use, particularly for Dr. Kiola's study on the polystyrene surfaces, we use cinnamaldehyde and scorin. So cinnamaldehyde is already one of the major active component derived from cinnamon oil. So we already use a concentrated ingredient as compared to using a oil that combines with all kinds of the ingredients. So we have already used the most effective active compounds as a phytochemical to control biofilms.
Okay, thank you. One more question. I know you discussed 3 different types of technologies. Um, can you compare them, cost-wise, economically, how do they compare each groups of these technologies for biofilm production?
Okay, so I'm not like a super expert here, so please correct me if you have like more answers or something. I'm happy to open for discussion. But for my opinion, I would think like the nano emulsion it will be the most cost-effective method because as I mentioned, that only requires water, oil, and a good working surfactant. And it is also very easy to be synthesized like you just need to, for instance, for the high energy method, you just need to have like an ultrasonificator where when the composition are in the correct ratio, they will form a very stable nano emulsion for a very long time. So as compared to 2 other technologies, I would think that the nano emulsions will be the most cost-effective method.
Okay, thank you. Does anybody have any questions? You can unmute yourself and you can ask the question as well. Yeah, I do see a question from Norm Barnett at the Arizona Department of Ag. What is your best advice to those currently in the ag industry that clean and sanitize harvesting equipment in the field to avoid biofilm buildup? That's a great question.
That's a great question. So as I mentioned, like you have to think about what is the condition of the food processing facilities. So if you want to claim the like a washing tank that have high humidity and high moisture, you want to pay more attention, making sure that every single corner has been extensively sanitized, avoid the bacteria survive in the biofilms. So I would say it depends on what type of the equipment you are working on. So some of the studies, like the peptide study we have to, we specifically want to use that solution to be used for sanitizing the equipment, like the instrument or tools. So they can be soaked into the peptide solution for up to 24 hours and then effectively to remove the biofilms. So depending on your infrastructure or your equipment type, you will use different type of the method to control the biofilm formation. I hope that answered the questions.
Thank you. Does anybody have any other questions? Okay. If nobody has any other questions, maybe give it 1 more minute. So one more question for you, Amy, while we are waiting. I know you talked about mixed biofilms, wherein you talked about, two different types, like mixed layer, and then, even layers of, each type of bacteria. Have studies been done on these mixed biofilms, and have they found, one type of mixed biofilm is easier to eradicate than others? I mean, is there anything done on those?
So, like I said, it's very complicated to be studied. So most of the study we came across is that some part of the multi-species biofilms is using something like CDC bioreactor. In turns, one of our PhD students is also conducting this type of the research using Ralstonia insidiosa with some other pathogens. And we enumerate the bacterial populations for each bacterial species to see, compare with the single species biofilm to see that if total amount of the populations has been increased in the presence of the non-pathogenic bacteria. Other type of the research also using microscope, like microscopy images, so they could have different targeted fluorescence for each different bacteria, and then they let them form biofilms on the glass slide. And once they read under the microscope, they can, using the three-dimensional method to determine the organization pattern they have.
Thank you. Any other questions from anybody? All right, yeah, I don't see any more questions. I would like to thank our speaker, Dr. Yin, for giving the presentation today. It was a wonderful presentation. I'm sure, we will find it beneficial. I would like to thank all of you for joining us today. And I would like to thank Debbie Reed for setting up the Zoom and organizing this seminar today. So if someone missed it, there would be a recording of the presentation at the Food Safety Consortium website, so you can watch it later. I hope everyone has a wonderful summer, and I hope to see some of you or all of you at the next Food Safety Consortium monthly meeting on August 24th between 2 and 3 p.m. Thank you again.